Blood is the vital fluid that circulates in our veins. Let's get to know this life-giving fluid a little closer, which is always with us. Blood is the red-colored, vital fluid that carries oxygen, nutrients, hormones, vitamins and antibodies to the tissues, and removes carbon dioxide and waste materials from the body. Blood is called hema in Latin, and the branch of science that studies blood is called hematology. While blood is so vital, it goes without saying how important human-to-human blood transfusions are. The first attempts of blood transfer are based on a rather tragicomic story. In the 16th century, sheep blood was started to be given to people who caused problems in order to make them docile like sheep. You heard right, sheep blood transfers have been done. What a wonderful theory, isn't it? However, when this situation did not give good results, human-to-human blood exchange trials were started. Although when there are too many deaths during human-to-human blood transfer, it has been discovered that there are other factors to be considered here. Karl Landsteiner, an Austrian-American immunologist and pathologist of Jewish origin, worked as a researcher at the Vienna Pathology Institute between 1898 to 1908. In their work, by finding differences in the structure of blood in humans, they revealed the dangers of improper blood transfusions. In 1901, he showed that there are at least 3 blood types in humans, according to the type of antigens that bind to the outer layer of the cell membrane in red blood cells. He named these groups A, B, and O. A year later, group AB was found, which had both A and B antigens and no AB antibodies. Landsteiner detected the M and N groups in 1927, and the rhesus factor, named after the monkey species, in which it was first found, in 1940. Rh factor is a difference that can occur in the blood of the mother and fetus. In addition, this difference was the basis of a series of reactions that led to miscarriage, stillbirth, or a fatal illness in the newborn. Thanks to these biological developments, it was also used in medical practice, and blood was transfused without endangering the patient's life. Landsteiner's work contributed significantly to the development of forensic medicine, enabled blood types to be used as reliable evidence in paternity and murder cases. If we go into detail about why ABO blood grouping is important, when examined under the microscope, we can see that it contains antibodies against the antigen, which are formed without encountering any antigen in the ABO system depending on our blood group. For example, an individual with type A blood has anti-B antibodies. If B blood group blood is transferred to this person, the blood will be hemolysis and result in the death of the patient. You heard right, blood transfusions are that vital. The branch of science that studies these blood transfers is called immunohematology. What are the blood group determination methods applied before transfusion procedures, which are of vital importance? Blood group determination processes are performed by using slide, tube, gel centrifugation and microplate methods. The most basic method is hemoagglutination, so this means that agglomeration of blood. In unresolved cases, methods such as PCR are also used. We do not touch on what antibody and antigen mean, because we have explained it in detail in our video on the subject. Hey, don't worry if you forgot, let's remember by watching it again and come back here to digest more.